We're going to be investigating fruit juice, egg whites, starch, regular cooking oil, and we're also going to use distilled water, etc. Let's start with the iodine test, which is the test for starch. First, take your iodine solution and add it dropwise to each of your samples. Notice that the iodine is a kind of orangey colour, and when we add it into our samples, that orangey colour remains, until we add it to the starch solution and there's a clear blue-black change in colour. We continue adding this to all of our samples, and amongst all of them, starch is the only one that shows a positive result. So to summarise, we use iodine to test for the presence of starch, test for the presence of proteins, add it dropwise to your samples, and it'll turn purple or lilac colour if protein is present. The Benedict's test is the test for reducing sugars. Glucose is an example of a reducing sugar. First, you take your Benedict's reagent and you add it dropwise to each of your samples. It's a good idea to label each of your samples for this test because you're going to need to put them into a water bath and they might get mixed up. For your water bath, there's a number of ways to do it, but the easiest way is to just boil the kettle and fill up a beaker with hot water. Put each of your samples into the water bath and then wait for a while. It does take some time for a reaction to occur, but if we watch this speeded up, you can see that one of our samples is clearly turning a kind of yellowy orange colour, and this indicates the presence of a reducing sugar. When we put them back into the test tube rack, we can see that fruit juice is the only one that's actually reacted to this, suggesting that it contains reducing sugars. So, to summarise, we use Benedict's reagent to test for the presence of reducing sugars. Add several drops to your samples and then put them into a water bath. Depending on how much reducing sugar is present, it'll first turn green, then yellow, then orange, and it could form a brick red precipitate if there is a lot of sugar present. The ethanol emulsion test is the test for the presence of fats. First, you need to measure out 2 centimeters cubed of ethanol and pour that into each of your samples. With each one, you need to shake it vigorously. After you've shaken your samples, add 2 centimeters cubed of water and then observe the changes. Here in our oil sample, you can see this sort of white cloudiness formed. This is what's known as the emulsion. To summarise, we use the ethanol emulsion test for the presence of fats. Add 2 centimeters cubed of ethanol and then shake, followed by 2 centimeters cubed of distilled water. A white emulsion forms if